So let's talk about first step of cellular respiration, the production of acetyl-CoA. Uh, the production of acetyl-CoA is a way for of bringing catabolizing different kinds of organic matter into this uniform two carbon pieces that could be subsequently oxidized to carbon dioxide and water. So there are different ways to generate acetyl-CoA. In this lecture, we will focus on a specific way how it is done from the end product of glycolytic pathway pyruvate. As we know, pyruvate is produced in the cytoplasm. So to make acetyl-CoA, it first has to be transferred into mitochondrial matrix through specific pyruvate channels called mitochondrial pyruvate carriers. So the production of acetyl-CoA is strategically placed within the mitochondrial matrix, which makes it practical to subsequently burn acetyl group into citric acid cycle and produce the reduced form electron transport carrier, which could be at the end burned on atmospheric oxygens through the electron transport chain sitting in the inner mitochondrial membrane. So why do we need to make acetyl-CoA in the first place? And the chemical reason is that there is terminal methyl group in pyruvate which cannot be easily oxidized. So it has to be transferred somewhere to some larger molecule, and the larger molecules could be chopped off at the end, at its ends, getting oxidized to CO2, and this way the methyl group could be indirectly oxidized. So this is the reason why to generate acetyl-CoA. And acetyl-CoA, within acetyl-CoA, the methyl group is getting activated sufficiently for to make it reasonable from thermodynamic perspective to transfer it to bind it to something else. And as we will see, this is something that happens in the first step of citric acid cycle. So what is coenzyme A and how the methyl group is getting activated by binding to coenzyme A? Coenzyme A is a chemical group carrier, or to be more specific, active acetyl carrier, which is a nucleotide based cofactor consisting of adenine nucleotide derivative with bound extension called pantothenic acid, which is also known as vitamin B5, and further extended by beta-mercaptoethylamine group. At the end of beta-mercaptoethylamine is this reactive thiol group. So, and at the end, when forming the acetyl-CoA represents a thioester. And if we would compare this thioester with normal oxygen-based ester, then while in normal esters, there is a way for pi electrons between two oxygen atoms to delocalize, thus stabilizing the compound. There is no such a way in thioester. This means that the free energy of the thioester as a compound as compared to normal ester is the energy content is significantly higher, and this is used as a means to destabilize this methyl group for easiness, to make it easier its further transfer to some other carrier, which we'll see later. So this is the chemical logic for producing acetyl-CoA. So the reaction of acetyl-CoA production is called oxidative decarboxylation. And in this reaction, the pyruvate loses its terminal carboxy group into CO2, and the acetyl chunk is getting transferred, forming the thioester with the reduced form of coenzyme A. Concomitantly, the redox cofactor NADH, NAD, sorry, gets reduced into NADH within this reaction. So altogether, the reaction is quite complex. It consumes pyruvate, reduced form of coenzyme A, oxidized form of NAD plus, and outputs the reduced equivalents in the form of NADH, CO2, and then esterified, TO-esterified form of coenzyme A with acetyl group, acetyl-CoA. In total, in spite of the fact that the thioester of acetyl group is a highly energetic molecule, this is a very highly exergonic process. So it is virtually an irreversible reaction in the cell. And the reaction is, as we already see, is quite complex, and it consists chemically from three different stages. In the first stage, pyruvate is decarboxylate, losing its terminal carboxy group, and the respective acetyl carbanion secondly loses two electrons in the process of oxidation, and then gets transferred, esterified with acetyl CoA, coenzyme A, converting this to final product acetyl CoA. So each of these reactions is actually catalyzed by separate enzyme, which are called pyruvate dehydrogenase, dehydroxylipyl transacetylase, and dehydroxylipyl dehydrogenase. And each of these enzymes, they are abbreviated E1, E2, and E3, and actually different. These are different polypeptide chains, which are all together present in one machinery, in one complex, with different stoichiometry of different of the different E1 and 2 and 3 enzymes, which are all together called pyruvate dehydrogenase complex. And to understand why it is so big and so complex, we have to look at the inner workings of the pyruvate dehydrogenase complex. This is a schematic representation of PDH complex. In these schematics, the E1, E2, and E3 activities are represented as shown in yellow, green, and pink. So the function of PDH complex could be compared to a factory line where the substrates and intermediates and the products and electrons are transferred between these enzymatic activities akin to the products in the assembly line between the stations through the active group carriers and electron carriers as robotic arms. So the process starts with the E1 subunit, E1 activity accepting the substrate pyruvate. The function of E1 could be is very similar to the function of pyruvate decarboxylase when we talked about ethanol fermentation. There, pyruvate decarboxylase was used in thiamine pyrophosphate to decarboxylate pyruvate and form active acetaldehyde, which was then released into simple acetaldehyde into solution. Here, very similar thing happens, except that the intermediate hydroxyethyl thiamine pyrophosphate is not decomposed, collapsing into thiamine pyrophosphate and acetaldehyde, but this um, bound hydroxyethyl group to thiamine pyrophosphate is transferred to E2 subunits to reduce the uh, lipolysin group carrier from its oxidized form to reduced form. And lipolysin is this the lipayl sulfur compound which is bound to lysine within the active center of E2. And hydroxyethyl, when transferred from thiamine pyrophosphate to oxidized lipolysin, reduces it to its reduced state, reducing the disulfide bond and forming one of the tile groups and one branch, another sulfur atom forms the thioester with the acyl remnant of the pyruvate. This way, oxidizing the aldehyde form into carboxy form in essence. Then this semi-reduced lipolysin intermediate is esterified with acyl group, is used to transfer acyl group to acetyl-CoA in a process called transesterification, and this way the acetyl-CoA as a final product is generated, and concomitantly the fully reduced form of lipolysin is formed. The reduced form of lipolysin is then regenerated back to an oxidized state by transferred electrons through the E3 subunits on NADH. And then in 3, what happens is that the FAD, which is an integral part of E3, is used as a electron transport carrier to oxidize the lipolysin into its oxidized state and concomitant transfer, reducing equivalence, forming the reduced form of FAD5H2, which is then on the 3 is regenerated back to its oxidized state by NAD plus as an electron transport carrier, reducing it to NADH. So this way, whole process is really a set of very tightly physically and chemically linked reaction where the substrate and intermediates are hopping between these 3 enzymatic activities as stations within the production line. So the first again decarboxylation is happening on E1. So then the intermediate is used to reduce the lipolysin, transferring 2 electrons, then transacetylation occurs or transesterification occurs, producing acetyl-CoA, and then the FAD is used to regenerate the lipayl group, and finally the electrons released from this redox reaction are transferred, evacuated through NADH. So this way, the steps, because of the tight physical linkages between the enzymes forming one complex, can occur extremely fast, and this is the mode of action is called the substrate channeling. The substrate channeling is a very general principles and is found in other enzymatic complexes beyond the PDH complex, allowing various metabolic processes to occur much faster and much more efficient. So altogether, the one could appreciate the complexity of pyruvate dehydrogenase complex, consisting of 3 different enzymatic activities and 5 different cofactors, uh, of which 2 are the electron transport carriers, NAD and FAD, and 3 active group carriers, thiamine pyrophosphate, lipoate, and coenzyme A. As an interesting fact, the activity of PDH complex consists of requires 4 vitamins. NAD and FAD electron transport carriers such are dependent on the vitamin B3 and B2 respectively, niacin and riboflavin, and 2 out of 3 chemical group carriers, thiamine pyrophosphate and coenzyme A, they production in our organism is dependent on the supplementation with respectively vitamin B1 thiamine and vitamin B5 pantothenic acid. So the last question is why to form such a big complex with different stoichiometry of the enzymes. The outfit of PDH complex consists of the E1 subunits which encircle the central part consisting of E2 and E3, and as such within PDH complex the E1 is present in the stoichiometrically high amount as compared to E2 and E3, and the reason for this is that the E1 is much lower enzymes as compared to E2 and E3, and having such a big complex containing much higher stoichiometry of E1 as compared to the rest makes the metabolic flux between within this chain of reaction functioning optimally, where slowness of E1 is compensated by its higher number of the subunits. But before wrapping up with PDH complex, let's look at the milestones of this activity. In the first step, the decarboxylation of pyruvate takes place. And at the heart of this reaction chemistry is the diazolium ring of thiamine pyrophosphate that allows to delocalize electrons and transiently stabilize carbanion forming in this reaction that at the end allows to release CO2 and form this covalent intermediate hydroxyethyl thiamine pyrophosphate. At the second reaction, in the second step, the the hydroxyethyl pyruvate is used to reduce the oxidized form of lipolysin in the heart of E2 active center, thus performing 2 processes, transferring electrons and transferring acyl group. So the lipolysin is an interesting compound. So it is lysin within active center of E2 with covalent extension lipoic acid, which altogether represents this very long extended molecule, and its extended nature allows to physically reach the E1 site from E2, thus allowing the physical transfer of hydroxyethyl from thiamine pyrophosphate to lipoate. In its oxidized state, the lipoic acid is a circular molecule which is circularized by this disulfide bond. And the fact that this disulfide bond allows lipoate to work as a versatile carrier, at the same time carrying reducing equivalence electrons and the acyl group. Being reduced by the by hydroxyethyl group transferred from thiamine pyrophosphate, the reduction of the disulfide bond allows it to carry hydrogen on one branch and acyl group on the other branch. At the third step, or second part of the second step, the the transesterification activity within E3 allows to transfer the acyl group from the reduced lipoate to coenzyme A, thus generating the final product, acetyl coenzyme A, and completely reduced form of lipoate, dehydrolipoate. And the final step of the reaction is regeneration of lipoate in its oxidized form from dehydrolipoate using E3 activity and FAD as an intermediate carrier of electrons that, due to the activity of E3, end up in the final electron acceptor NAD+, which is reduced to NADH and thus evacuates electrons or reduced equivalents from the system. And this is all about the PDH complex activity and its inner workings.